Hello everyone, this is Mohammad Imdadullah. So in this video lecture project, I will discuss about the enzyme phosphofructokinase. So in this lecture, I will discuss about the overall structure of the protein. I will explain what is important. Then in the next sections, I will discuss the catalytic activity of the proteins and go into detail into the active site morphology. At the last, I will discuss about the regulation of the protein and discuss how the activity is modulated, turn on or turn off. Then at the end, I will discuss about the clinical significance of the enzyme phosphofructokinase. So let's begin. That first, we need to know what is an enzyme. So an enzyme is a biological catalyst and is almost always a protein. It speeds up the rate of a specific chemical reactions in the cell. For enzymes, the enzyme is not destroyed during the reaction and is used over and over. So now in the figure, you can see the mechanism of the enzyme activity. Here you can see the enzyme and the substrate bind to the active site. Then an enzyme and substrate complex is formed, and then at the end, the enzyme produces the products. So this is a basic, very general overview of how an enzyme works. So we'll next go to the next slide. Now we'll discuss. Now we'll give a short overview of the introduction to the phosphofructokinase 1. So phosphofructokinase 1 is an important glycolysis regulatory enzyme. It is an allosteric enzyme with 4 subunits that is regulated by numerous activators and inhibitors. The important commutative step of glycolysis, the conversion of fructose 6-phosphate and the ATP to fructose 1,6-bisphosphate and ADP, is catalyzed by phosphofructokinase 1. Phosphofructokinase can regulate glycolysis, allowing the cell to increase or decrease the rate of glycolysis in response to the cell's energy requirement. A high ATP-ADP ratio will inhibit phosphofructokinase and glycolysis. Another thing is that phosphofructokinase is activated by fructose 2,6-bisphosphate. So another you can see a high ATP to ADP ratio, for example, will inhibit the phosphofructokinase and glycolysis. The primary distinction between the phosphofructokinase regulations in eukaryotes and prokaryotes is that phosphofructokinase in eukaryotes is activated by fructose 2,6-bisphosphate, and the purpose of the fructose 2,6-bisphosphate is to replace ATP inhibition, allowing the eukaryotes to be more sensitive to hormone regulation by glucagon and insulin. Now, here we can see the structure, the tetrameric structures of the phosphofructokinase. Here in the 4 different colors show the 4 units, and this all together form the tetrameric structure. So we can see the mammalian phosphofructokinase is 340 kilodalton tetramer composed of different combination of 3 types of subunits. The 3 types of subunits: the muscle, liver, and the platelet. The composition of the phosphofructokinase differs according to the tissue type it presents in. For example, mature muscle expresses only the M isozymes. Therefore, the muscle phosphofructokinase 1 is composed solely of homotetramer of M4. The liver and kidneys express predominantly the L isoform. In erythrocytes, both M and L subunits randomly tetramerase to form M4, L4, and 3 hybrid forms of the enzymes. So, each subunit of the tetramer is 319 amino acids and consists of 2 domains, one that binds the substrate ATP and the other that binds the fructose 6-phosphate. The N-terminal domain has a catalytic role binding the ATP, and the C-terminal has a regulatory role. So you can see the phosphofructokinase 1 belongs to the family of phosphotransferase, and it catalyzes the transfer of gamma phosphate from ATP to fructose 6-phosphate. The phosphofructokinase 1 active site comprises both the ATP magnesium ion and the F6P, fructose 6-phosphate binding sites. Phosphofructokinase 1 is an allosteric enzyme whose activity can be described using the symmetry model of allosterism, whereby there is a concerted transition from an enzymatically inactive T state to the active R state. For every molecule of phosphorus 6-phosphate that binds to phosphofructokinase 1, the enzyme progressively shifts from T state to the R state. A graph plotting phosphofructokinase 1 activity against increasing phosphofructose 6 concentration would adopt the sigmoidal curve shape traditionally associated with the allosteric enzyme. So here we can see a graph plotting the PFK-1 activity against increasing F6 concentrations would adopt the sigmoidal curve shape traditionally associated with the allosteric enzymes. Allosteric activators such as AMP and ADP. Inhibitors such as ATP and PAP bind to the some allosteric site and facilitate the formation of the T state, thereby inhibiting the enzyme activity. You can see phosphatokinase activity and the fructose 6-phosphate concentrations, high ATP concentrations, and the low ATP concentrations. So when there is a low ATP concentrations, the fructose 6-phosphate, the phosphatokinase activity is much higher, and when there is a less high ATP concentration, the phosphatokinase activity is much lower. So here in the figure, we can see the catalytic activity, how the fructose 6-phosphate react with an ATP, and the ATP converted to the ADP, and it results in the formation of fructose 1,6 bisphosphate, fructose 6 phosphate. Well, the most important thing, the regulation of the phosphatokinase. So phosphatidylkinase 1 is the most important control site in the mammalian glycolytic pathway. The first irreversible reaction is unique to the glycolytic pathway. This leads to precise control of glucose and the other monosaccharides, galactose and fructose going down the glycolytic pathway. Phosphatidylkinase 1 has two sites with different binding affinities for ATP, which is both a substrate and an inhibitor. Phosphatidylkinase activity is reduced through repression of synthesized by glucagon. Glucagon activates protein kinase A, which in turn shuts off the kinase activity of PFK2. This reverses any synthesis of F26 bisphosphate from F6P and thus deactivated phosphatidylkinase 1. The precise regulation of phosphatidylkinase 1 prevents glycolysis and gluconeogenesis from occurring simultaneously. Serotonin increases the phosphatidylkinase by binding to the 5-HT receptor, causing the tyrosine residues of phosphatidylkinase to be phosphorylated by a phospholipase C. Where in the figure we can see the regulation of phosphatidylkinase 1. Phosphatidylkinase 1 is allosterically inhibited by high levels of ATP, but AMP reverses the inhibitory action of AMP. The activity of the enzyme increases when the cellular ATP to AMP ratio is lowered. Phosphatidylkinase 1 is also inhibited by low pH levels, which augment the inhibitory effect of ATP. Finally, phosphatidylkinase 1 is allosterically inhibited by the PEP citrate and ATP. So in the figure, we can see what I have discussed right now. Now let's discuss about the clinical significance. So a genetic mutation in the phosphofructokinase AIM gene result in the Tauris disease, which is a glycogen storage disease, where the ability of certain cell types to utilize carbohydrates as a source of energy is impaired. ATP is a natural allosteric inhibitor of PFK in order to prevent unnecessary production of ATP through glycolysis. However, a mutation in ASP543L can result in ATP having a stronger inhibitory effect due to increased binding to PFK's inhibitory allosteric binding site. So again, in order in cancer, how PFK play a role in cancer? So in order for cancer cell to meet the energy requirements due to the rapid cell growth and division, they survive more effectively when they have a hyperactive PFK-1 enzyme. When cancer cells grow and divide quickly, they initially do not have as much blood supply and can thus have hypoxia. And this triggers O-glucose anisylation at serine 529 of PFK. This modification inhibits PFK1 activity and supports cancer proliferation, in contrast with the view that high PFK1 activity is necessary for cancer. This may be due to the redirecting glucose flux towards the pentose phosphate pathway to generate NADPH to detoxify reactive oxygen species. At last, we can see some viruses, including HIV, ACMV, and Maillard effect, cellular metabolic pathway, such as glycolysis by MOI dependent increase in the activity of PFK. The mechanism that herpes increases PFK activity is by phosphorylating the enzyme at the serine residue. The HSV1 induced glycolysis increases ATP content, which is critical for the virus replication. So we can say that the phosphatidylkinase 1 has a great importance both in the clinical significance and also have a very important role in the significance of various diseases. They have many important significance in clinical sites. So we should know about the phosphatidylkinase enzyme 1 and we should study and have further research on this field. So that's with I want to conclude my speech. Thank you. Thank you all.